me do that. All right, perfect. And then for folks who are interested, the next step in that process would be that there's actually a competition for funded projects. So two or more junior investigators, they could be from this group of people. So it could be one of the four of you, or it could be one of you or two of you with people from other groups, can come up with a collaborative project idea. And then you can actually submit that to IEM and basically compete for funding on your collaborative project. And so those are going to be due October 12th. So that's 2 weeks. There's more details at this link here. I think you guys are going to receive all this information independently, but just to show it to you here quickly. So hopefully some of you will be interested in this and take advantage of it, because I think it's a cool opportunity. Okay, any questions about what we're going to do here? All right, let's get started. So I'm going to stop my share and I'm going to make it so you guys can share your screens. And let's start with Minhan. You good to go first, Minhan?
Sorry, I'm on mute. Okay. Yeah. Can everyone see my screen now?
Yep.
Okay, perfect. Okay, I will begin now. Hi, I'm Minghan from Department of Biomedical Engineering. So today the topic of my poster is characterization of antigen presentation and T-cell response in different focal therapeutic conditions for immune priming. In recent years, there is growing evidence that minimally invasive cancer focal therapies, including thermal therapy, cryosurgery, and irreversible electroporation, can engage the immune system for systemic and long-term disease control. However, the underlying mechanism remains unclear. Based on our previous study using B16 model, here we continue our investigation using a model antigen, OVA, to further study important factors in focal therapies, including antigen release, protein denaturation, and damage-associated molecular patterns. This figure showed our in vitro antigen presentation assay and T-cell assay. The dendritic cells are purified from bone marrow, differentiated with FIT3 into antigen-presenting cells, then pulsed with focal therapy-treated B16 OVA cell lysates. Then we will use flow cytometry to characterize the antigen presentation by quantifying the percentage of MHC1 symphycal complex. By looking at the T cells, we will purify the OVA-specific T cells from transgenic mice, then label them with a violet dye. The proliferation of T cell will be characterized by the dilution of dye, while the activation of T cells will be detected by the expression of specific activation markers. This figure shows the proliferation of OVA-specific T cells after different focal therapy conditions. We can notice that the T-cell response after the IV treatment is more robust than both cryo and heat. So what caused the difference in T-cell response? In B16 OVA lysates, there are antigens and danger signals. So our study is focused on these two elements. First, we look at the influence of focal therapies on pure antigens. This representative Western blot image shows that showed the relative quantity of a released OVA from B16 OVA cells after different focal therapies. This preliminary result suggests that the method of focal therapy can actually affect the amount of antigens released from treated tumor cells. Next, extreme focal therapeutic conditions were applied to OVA protein or peptide to see how focal therapy influences the immune processing on the level of pure antigen. These two figures showed the antigen presentation of OVA peptide and OVA protein separately, which is time and dose dependent, while this figure showed the presentation of focal therapy treated OVA peptide symphycal. Actually, the presentation of Synfecal alone were relatively unaffected by focal therapy type under the conditions we tested. We'll also look at the T-cell response by characterizing specific activation markers, including upregulation of CD69 and CD44, and downregulation of CD62L. From this data, we can see that the focal therapy conditions on pure protein without involving cells don't contribute to significant change in dendritic cell or T-cell response. In contrast, the T-cell priming is very sensitive to the quantity of antigen. In addition to antigen, DAMPs is also an important element in cancer immunity. We propose that the method of focal therapy can influence the DAMP release profile and induce differential T-cell response. Here, the B16-OVA cell lysates are analyzed for cytokine, HMGB1, and ATP release by cytokine array, ELISA, and luminescence assay respectively. Then to study how DAMPs influence T-cell priming when the antigen is the same. The dendritic cells are pulsed with naive ova plus focal therapy treated B16 lysates. With pure protein, the T-cell activation is quite limited, but when you add B16 lysates, we can see significant increase of T-cell activation markers. This observation may suggest an important role of modulating immune priming by DAMPs, and more data will be collected to compare between different treatments. For lysate characterization, we scanned 25 cytokines and select some that we think might be important.
Minhan, how much more do you have? We're at time here. So.
Just this one, just this one.
Okay.
Yeah. So we select some that might be important in this process. Here, the IP10, ATP, and HMGB1 are considered as immune promoting, while interleukin 10 is a major immunosuppressive cytokine. This preliminary data suggests that the focal therapy induced differential DAMP release profile, which contribute to distinct immune response. Yeah. So generally, our work expanded the knowledge of immunomodulation by focal therapy, and it also serves the basis of optimizing combinatory strategy with immunotherapy, for example, immune checkpoint inhibitor to address the local recurrence and metastasis. Yeah, thank you for listening. Yeah, that's all.
Okay, so I have a couple questions. So
Sure.
Can you go back to slide 2, I think? This one, that one.
This one?
Yeah,
okay.
No, previous one.
Oh, previous.
Oh, no, next one, I'm sorry.
Oh, yeah.
Yeah,
no
there
problem.
we go, thank you.
Yeah.
So what do you think is going on with the heat here? Because it seems to be quite, and it doesn't appear to be doing a lot in this particular one.
Mm-hmm. Yes, so here we think that the actually heat lead to large extent of protein denaturation, which might prevent the exposure of the epitope of our model antigen. Here is the OVA protein, and yeah, and also the protein aggregates might also prevent the antigen processing by the dendritic cells. So that's what we think might dampen the immune priming by the heat-based focal treatments.
Okay, and then really quickly, so related on your last data slide, it looks like you have quite a different response of these various cytokines to the different treatments. So what do you think that might actually do? What do you think the biological result of that might be?
Oh, yeah, sure. Actually, here we use a cytokine array that include 25 cytokines, and we pick some that with significant difference here because we want to explain why the array is better than other treatments. So here we pick some, for example, the IP10 is a cytokine that we think might be important for leading the array treatment. So if this is really the key cytokine in the IRE treatment, then for the in vivo translating work, we might consider that inject the IP10 as an immune adjuvant to help with the focal therapy, then to improve the immune response after focal treatment. That's the biological relevance that we can consider for our project.
Okay, thanks, Manhan. Okay, Gaiden, are you good to go next?
Sure. Are you able to see my slide?
Yep.
All right. Hi, everyone. My name is Gaida. I'm a graduate student in David Odie's lab at the University of Minnesota. Today I'll be talking to you about some of my PhD work and understanding cancer cell migration in the context of glioblastoma. Glioblastoma is a brain tumor. It's one of the most prevalent, most common malignant brain tumors with really low survival. A patient diagnosed with glioblastoma has a 15 months median survivor and 5 years survival outcome of less than 5%. This MRI sequence shows the progression of the disease and how rapid the progression of the disease. Patient diagnosed with glioblastoma undergoes surgery only, and which the majority of the cases, patients the tumor reoccur. In this case, 3 months after surgery, the tumor reoccurs, and 2 months after that MRI was taken, you can see how far the tumor has spread. This also could be demonstrated here with this histology images showing the infiltrative nature of these tumor cells. So we previously have shown that CD44 can predict cancer cell migration and predict survival. In mouse models, we have shown that elimination of CD44 can reduce cancer cell migration, glioma cell migration, and improve survival relative to the wild type. However, when you overexpress CD44, you start decreasing the amount of migration in cancer cells, and that improves survival. This was also translatable into the human disease, where we have shown that different amount of CD44 transcript can correlate with survival in a biphasic manner. We have a minimum in survival with a median expression of CD44. And there was also association with subtype, which saw that proneural subtypes tend to have lower amount of CD44 expression, and mesenchymal subtype has the higher CD44 that really line up in the middle. So this kind of raised the hypothesis that a key mechanistic difference between GBM molecular subtype is that proneural cells are slow migrating and mesenchymal cells are fast migrating. To test this hypothesis, we actually developed, in collaboration with Dr. David Largaspada's lab, we developed mouse models to model both the mesenchymal and proneural subtype. We used RNA sequencing to validate that these models are able to capture human glioblastoma transcriptomic changes using cross-species analysis. We showed that NRAS tumors are elevating transcript that are commonly elevated in mesenchymal tumors, human mesenchymal tumors, and PGF beta driven tumors are has an elevation of transcript that are commonly overexpressed in proneural subtype human glioblastoma tumors. And in addition to the subtypes, we're also looking at the migration genes that are changing in these tumors. And we, to our surprise, only 4 genes were elevated in these. That are elevated in the. They are differentially expressed, and these 4 genes were all related to adhesion molecules, and they're all elevated in the NRAS tumors the one that models them as mesenchymal subtypes. So using our cell migration simulator model, we simulated the different scenarios, and we predicted that the NRAS mesenchymal tumors will have a higher migration relative to the low adhesion, the PGF beta proneural subtype. We were able to test these prediction using ex vivo imaging, where we can image brain slices, tumor-bearing brain slices, and we're able to quantify these migration. And we showed that the migration was enhanced in NRAS tumors as predicted relative to the PGF tumors. And we showed this is also independent of the tumor microenvironment because we isolate these cells and put them in healthy brain slices, and we still see the same difference in migration. And we also show that this is relevant to the human disease using Bayesian-derived xenograft lines. We acquired from the Mayo Clinic. We are able to test that mesenchymal tumor cells do indeed have faster migration relative to the proneural tumor cells. As predicted also by our modeling, there are increase in traction forces generated by mesenchymal tumor cells, which makes sense. If you increase the amount of adhesions in the tumor cells, they will start generating higher forces. And this is something we also were able to quantify using traction force microscopy. Kind of in interest of time, we also looked at I'll go quickly through this. We also look at the survival of these animals. We would predict that NRAS tumor cells would have a worse outcome, but to our surprise, that was not the case. but we also were able to attribute this to a difference in immune response in these tumor models. We showed that similar to human disease, NRAS tumor cells have elevated amount of immune response and activation relative to the BGF tumors, and we also validated that using IHC staining. So in summary, we developed tumor models that are able to recapitulate the two subtypes of human glioblastoma, and we were identified two characteristics. One, that mesenchymal NRAS tumors have faster migration relative to PGF for neural subtype, and they have a different amount of traction forces being generated. We also established an integrated biophysical modeling and experimental approach to mechanically parameterize and simulate distinct molecular subtype of preclinical mouse models. So with that, I will take questions in this acknowledgement.
Thanks, Guy. A couple quick questions. So one is, so you show this increase in the CD44 leads to increased migration, which was your prediction. And so I know previously your group has shown that the adhesion, there's an optimality for that too, for migration speed. And so this would suggest that you must be moving towards the optimal and not away from it, correct?
That's correct, yes. So if you look at the, so we're using similar model to our previous work, which we don't actually add CD44 into the tumor model. So we are operating in this wild type scenario. So we are really at the minimum. And if you look at the human data, the mesenchymal subtype also lies right at the minimum of survival, which we would predict that that's also at the peak of the migration.
Mm-hmm.
So that's where I'm operating. That's
yeah, okay, that makes sense. And then the other thing is it possible to just overexpress the CD44 in the same cells so that you get them to show the response that you expect, as opposed to two genetically distinct cell types?
Are you saying in the NRAS models or the PGF models? Is that right?
Right. So to actually change the expression in one of those models so that you mimic the behavior you see in the other model.
For sure. So so in the NRAS model, which this is actually this data in the left right here, this is done in an NRAS model. So this was already done in here, where we manipulate CD44 levels and you can get this behavior. We definitely want to try it actually in the other models. Now we can increase it in the PGF and see if we can increase the migration and lower the survival. And this is something that is ongoing right now with other lab mates that are in their project. So we're trying to do that.
Cool. Thank you. That's great. Pagao, would you can go next?
Okay. Can you see my screen now? All right. So this is Pega. I'm going to talk about my project on characterizing miniature probes for preclinical cancer focal therapies. So if you're not familiar with the word focal therapies, there are some methods by which you can destruct undesirable tissues locally. And there are lots of researches going on around how they can be used to treat lots of diseases such as cancer. But there is one big problem with all of those researches. They use clinical size probes, which I'm showing here on the scale, for their preclinical research on mice. So as you can see in this picture, if we use this big size probe for a small mouse, we can expect inconsistency in the experiments, lack of control in probe positioning, unclear energy field distribution. So we've seen this gap, and we went ahead and designed our own miniature probes, which includes a cryoprobe, which freezes the tissue, a heating probe, which heats the tissue, and an IRE probe, which destructs the tissue by some ultra short, strong electrical pulses. And after doing that, we went ahead and characterized them in vitro and then in vivo to see what is the energy field distribution around our probes. So to begin with, I'm going to describe our cryoprobe characterization. So we started putting our probe into a tissue mimicking gel, and we put some thermocouples around the probe to just measure the temperature over time. And then we compared it to our simulated, and then we simulated the exact same case, and then we compared the energy field distribution to our experiments. And as you can see, there is a perfect match between our temperature time profiles at different locations. And then we're planning to just do this in vivo and put the probe inside a rodent hind limb tumor and just measure the temperature at the edge and do some IR imaging. And again, compare it to our simulated case, which is simulated in console multiphysics, to see if we're capturing the energy field right. To characterize our heating probe, we put the heating probe in a porcine liver samples, and then we put some thermocouples around inside the tissue around our probe. And again, we did the same thing, measured the temperature time profiles at different locations, and then again, compared it to our simulations, which is done in console. And there's less than 5 degrees Celsius error in our in comparison of our experiments with our numerical model, but we're working to get a better match. And for our in vivo experiments, we're doing the same as we did in the cryo, and we're measuring the temperature and taking IR images and then comparing it into our simulations. So for our IRE probe characterization, we are trying to just measure the current passing through a porcine liver samples that is sandwiched between the electrodes of our IRE probe. And then we're measuring the current we're measuring the current and we're comparing it to our simulated case. So in our simulated case, we're just applying the sigma and electrical conductivity values of porcine liver samples found in the literature. And this gives me these two curves that we can see that our experimental measurements just fits perfectly in our area of expectance. And for our in vivo modeling, we're trying to just perform IRE for different voltages, but for 1 Hertz frequency, 99 pulses over, again, a hind limb mouse tumor, and just measure the current just the same, and then compare it to our simulated case in console again. So to put it in a nutshell, we just designed and tested some measure probes for preclinical use on rodents, and then we characterized them. And the cryoprobe characterization, in the cryoprobe characterization, we were able to capture less than 0.5 millimeters difference in isothermal locations and less than 5 degrees Celsius in thermocouples measurements. And we have less than 5 degrees Celsius error, again, in heating probe characterization, and our IRE probe characterization is in really good agreement, as you saw on the previous slide. And we are also planning to do a patient-specific modeling to be able to capture the geometry and the probe placement in our in vivo, in all of the in vivo cases, because the geometry of the tumor and all other steps are different each time. And so the work in progress is in vivo modeling, which is ongoing, and also getting a better match in our heating probe characterizations. Yeah, thank you so much for your attention, and I'm happy to take any questions.
Thanks, Pegah. That's really cool. So what's the sort of, what do you think are the biggest sources of error here that go into your various models?
Yeah, so there's difference. So for example, in my heating probe characterization, the placement of thermocouples precisely around the probe is the biggest source of error. So they're small. There can be positional error. They can be thermocouple error. They can be like conduction that is going through this spacer to my thermocouple. So we're just trying to minimize these errors to just get a better match. Or even there might be radiation from the probe that is affecting our thermocouples here. Or in our cryoprobe, there might be the properties of the tissue mimicking gel, or even thermal properties in the porcine liver samples that I'm finding in the literature or I'm measuring. And also, again, thermocouple positioning. Yeah.
Yeah, how hard is it to get the interface between your probe and the bio sample correct for the models?
You mean how hard is it to simulate it? Do you mean the simulations or?
Yeah.
Do you mean like the boundary conditions that I'm applying?
Yeah. Yeah, I mean, I'm just wondering how hard it is to capture that accurately, because I can imagine when you stick the probe in, you create some mechanical damage, and I don't know how much that will actually affect the measurement.
Yeah, exactly. So, so for the boundary conditions, I'm just attaching I'm just soldering a thermocouples on the probe, and just I'm capturing the temperature on the probe during the time, and I'm also just having the convective coefficient of the probe from the company that we're manufacturing it together. And I am applying it to my console model. And for the thermal damage that you're talking about, there is a paper that is actually taking the thermal damage into the account into the thermal properties. So I'm just applying that into my model to get it fixed. Yeah.
Okay, thank you so much. That's great. Kellen?
All right, just one second. I'll share my screen. And present that. Okay, so my name is Kellen. My thesis work is at the Center for Magnetic Resonance Research over by the football stadium. And then I've also been doing this quantitative imaging research with Dr. Chris Wilkie in radiation oncology. This work on predicting the cellular motility of glioblastomas is a collaborative effort with the David Odie Lab. They've provided the motility data that we'll see in a second. The problem is that glioblastoma is deadly almost uniformly, and it seems that every glioblastoma paper that you read begins with the same paragraph about survival rates and how treatment hasn't changed in 20 years. And the statistic that always sticks out to me is that the recurrence rate of glioblastomas is expected to nearly be 100%. And so one reason for this may be that that these tumors are growing and they're spreading and they're metastasizing across the brain. And so, as we saw from Guidance presentation, understanding the motility and the migration characteristics of these tumors may be extremely helpful. The problem for eventual clinical translation is that characterizing the motility characteristics is expensive, time-consuming, and difficult. And so this project is really seeking to answer the question is if we can non-invasively characterize the cellular motility of these tumors using something that all patients go through, which is MRI. There's a quick flow chart of the methods that we use. So a patient will present usually with headache, dizziness, something, aphasia, and they get MR imaging, in which a tumor shows up, and then they'll have that tumor resected. So here's where the two branches of this project split out. So on the imaging side, we take this preoperative T1 weighted contrast imaging, which really shows you nicely where the boundaries of the tumors are. And then what we're going to do is we're going to segment the tumor. And for this project, it's manually, but hopefully in the future, if we get more data, it will be automatic. And then we do feature extraction, which we take 107 quantitative features from this region of interest of the tumor. And those are related to things like the shape, the texture of the image, which are the second and third order statistical distributions of the voxel values, and the intensity, things like the mean, the kurtosis of the voxel values. And now on the cell side, the Odie Lab is taking these cells, putting them into dishes, and then recording them over a long time period in order to calculate the mean motility of these cells. And so for each patient that we have, that means like 120 cells of recordings. So now we are taking our features, and we do a feature selection process, which is essentially a random forest feature selection, which ranks the contributions of different features. And then we are attempting to use those features that are selected by this process to predict the motility of these tumor cells non-invasively. And we, surprisingly, because a lot of times these this type of quantitative feature analysis gives us nothing, but we really got some compelling results from this, where the root mean squared error between the predicted motility and the actual motility is 0.77 millimeters squared per minute. So, and the R squared value of that fit is 0.6, which tells me that we have a pretty strong correlation between some of our imaging features and the cellular motility of these tumors. And just as an aside, we limited the model to 4 different imaging features because our data set had about 30 patients. So that's to primarily prevent overfitting. To validate the model, I performed leave one out cross-validation, which is a procedure that trains the model over and over and over, but leaving one data point out, then to use that, the average of error on those left out data points to establish a generalization error. And this was slightly lower than our root mean squared error with the model totally trained. So that tells me that there are still some outliers that are contributing to the model. As far as residual values, they don't show much of a relationship. So that tells me that the model doesn't need to be any more complicated. And then we did permutation tests where the model's trained on scrambled data. And our model, shown by the red line here, outperforms in terms of the R squared value, where the distribution is way to the left, and the root mean squared error, which is way higher than our distribution. Some limitations of this: we had a small sample size, 30, so we'd love to get more data for that. The other limitation that I can think of is that glioblastomas are heterogeneous tumors, and we're capturing 120 cells from this. So whether or not that's truly characterizing the motility of the entire tumor is probably an open question. But to me, this is it's a compelling result that we can use voxel values from magnetic resonance imaging to characterize tumors on the cellular scale. And then potentially this could be used in the future to characterize tumors based on their motility, to stratify them into treatment groups or decide, give an estimate of prognosis or something like that.
Thanks, Kellen. So I'm just help me understand. So what in the end, your predictive model is based on 4 of the imaging parameters, and how are those used for the prediction? How are they in some sort of, is it like partial least squares, or just some linear combination of those 4, or is it something else that's the predictive?
Yeah, it's a linear regression model. We use the adaptive lasso method, which does us a dual service by estimating the coefficients and eliminating unnecessary variables. So there were 10 or so imaging features that we selected using the random forest feature selection, and then the adaptive lasso set the coefficients for 6 of those to 0. So then those 4 left over, we had confidence that those were representing a pretty strong contribution to the motility prediction.
And in going forward, in terms of the imaging, so would you imagine the segmentation would be done by machine learning or something like that?
Yeah, the most popular, they do deep learning UNet approaches to do the segmentation. And so when we get like hundreds of these, it would be very nice on my end to just have a deep learning model do it.
Cool, thank you. Okay, let's open it up. So I think maybe the place to start would be to see if you guys have questions for each other. I think that's probably a useful place to see if there's some connectivity here.
I got a question for Kellen. This is really cool stuff. I know I've seen it also before. Can this technique or method be applied also to look at other features of glioblastoma, like immune infiltration and things like that? Or has it been applied?
Yeah, so I think one of the cool things about this data set that we've used here is it's really the first time that features like this have been looked at for quantitative imaging features in glioblastoma. And so, yeah, if there was a, you need data to do it, like dozens of data sets, but if you have that for a large number of tumors, that are already classified into groups, then it is very straightforward to try and see if there are imaging features that could do that clustering automatically.
Yeah, because I'm thinking of the Ivy project at University of Washington, I think, or it's or the Allen Institute. I think they have a collection of MRI images of glioblastoma patients, and they also have sequences data that one could use to predict immune contribution in those tumors, and then use the MRI images to extract feature that could actually be then used to predict immune infiltration in these tumors and whether we ever need to go and sequence these patients now in the future. You can just predict it from MRI images. That's kind of cool.
Right. Yeah, that seems to be the real power of this technique, is to do the expensive lab bench stuff non-invasively.
Yeah.
Obviously, though, you have to do that lab bench stuff first, though, to get the data. But Gaiden, I had a question about one of the slides in your presentation. I think it was the second slide. It was showing a figure from a paper from 2017, and it had like just showing the biphasic motility characteristics for the proneural versus mesenchymal. And maybe you're familiar with this data set, maybe not, but are all those data points, those are individual tumors from humans that were treated here? Because then, no. Oh, okay.
No, that's actually from a published data set. The human data set, that's from a published data set.
Oh, okay. Okay.
Not from here.
Because if there was MRI data for when you see, or when I see things that are like already in clusters based on lab bench data, my eyes kind of perk up, because I was like, oh, if there's imaging available, we could throw it at a wall, see what sticks. And
yeah. I mean, that's what I was talking about, the IVGPM project. I think they do have MRI data, and they have sequencing data. They have everything, and it's publicly available, I think. So you might
that's definitely
check it out.
Yeah, worth a look.
So I had a couple of questions / thoughts. Was like, so some themes that I picked up here were there's a lot of modeling going on here, and I'm just wondering if there's a way to connect some of those things. Migration's another theme here, and then of course imaging. And so, so for instance, I'm wondering, Minhan is seeing differential responses of cytokines to her therapies and the T cells. And it seems to me you might be able to predict T cell migration patterns based on what those cytokine profiles look like. And so that would sort of relate back to some of Gaiden's work. And then from Pega's work, I could imagine, I know the ODI lab has been doing modeling where they look at, basically proliferation and migration growth of a tumor. And it seems to me you could look at that after treatment with one of these focal probes, and then ask, would it be sufficient to actually kill the tumor if you account for what's going to happen after it's done? Yeah, I don't know. Anyway, those are a couple thoughts I had, so I just throw that out and see if people want to discuss.
Yeah, I think that's really cool, like the Minhan and the different cytokine expression, how that affect tumor immune cells migration in their native environment.
So actually, we are considered about the T cell migration pattern before, but it's a little bit difficult to look at, because so for example, when you're doing the focal treatment for a mouse tumor model, and then the antigens is transferred to the lymph node, and the dendritic cells will take up it and then activate the T cells, and then T cells will migrate to the either to the tumor part or to the peripheral blood, and it's really hard to look at the migration pattern. But in addition to the migration part, I think our lab and Odit's lab, we are talking about the possible collaboration for the focal treatment to the glioblastoma. And actually our lab is doing some experiments on the glioblastoma cells where some in vitro cell tests to look at the sensitivity of those glioblastoma cells to the heat treatment or cryo treatment. And we are also actively looking at some potential in vivo animal model. It's hard to do the real glioblastoma model, but we can do some simple hint lymph tumor model and to see how the focal treatment work on the glioblastoma
I think
model.
Manhattan, like one cool thing we could look into potentially. I mean, you showed the CD44 in your immune cells as a marker for, I think it's some response. So like, it's really kind of interesting to look at CD44 or its role in immune cells. Is it its activation? Is it driven by its ability now to migrate and go to the lymph node and antigen present and then go back to the tumor site? And maybe like doing, 2-photon imaging in vivo, like in vivo imaging, like of the lymph node, or you're using the B16 model, which is on the skin, which makes really easy using 2-photon imaging. And look at that migration behavior too. That's kind of, I think that would be really cool.
Oh yeah, this will be a very good way to look at the tumor infiltrating lymphocytes and then to predict some possible treatment effect. Yeah, that will be very cool.
Is there any way to combine is there any way to use the MRI to back out some properties of the tissue that might predict the ability of immune cells to infiltrate?
I mean, I think you could certainly try. You're talking about immune cells within a tumor. Yeah, I mean, it's something that if you could quantify it on the benchtop, it's definitely something that you could see if those features are explanatory or not.
So I could jump in add something a little bit to that one. So one thing I've been interested in working with some of the folks potentially over at CMRR with high field magnets is looking at like MR elastography. Then to look at basically some of the mechanical tissue properties of the tissues. So right now, I don't think they've done it for brain over there, but they have started doing some of it for liver, which then could also influence like some of the other talks that were discussed here today, particularly like some of the microprobe ones.
Yeah, definitely. And I'm also thinking of something else because we're a heat transfer lab and I'm the heat transfer girl. I can just, I was thinking of different levels of influence of heat on the migrating cells. I don't know if they have any effect, but if there is some treatments with focal therapy for glioblastoma, we can just see how that level of using heat or using cold just influence the level of cells migrating around.
Yeah, that's definitely interesting. Yeah. There's a lot of therapies that are being used and they definitely could have an influence in cancer cell migration. Be worth exploring.
Yeah, it's interesting too, because I guess, one thing you could imagine doing would be, you know, if you've got a prediction for what the heat profile, the temperature profile looks like in the tumor, say with the heat probe, though this could be applied to the other probes too, but the heat probe in particular, I'm thinking of, you know, you could look at, say, you know, where are you going to get necrotic tissue? And you could look at actually diffusive transport of whatever's released from that necrotic tissue and how that might affect signaling. But then you can also start to look at things like what are the temperature ranges over which you might start to see stuff like HIF1 alpha expression go up. So now you've got the tumor cells responding to that. What does that do? Does it change their migratory profile or something else that you might be able to predict some response from? So you could probably even get a little bit into molecular mechanism because some of that is known for temperature control particularly.
Yeah, definitely.
And also, so in the context of focal treatment, so in clinic, laser ablation is used for the treatment of glioblastoma. So I'm wondering if there are any research papers look at the influence of laser ablation on the, for example, glioblastoma cell motility, or it may affect the stroma, and which will also influence the motility of the cancer cells. So I'm wondering if there are any research about it.
Yeah, I haven't came across any that I know.
Yeah, I see some just preclinical study or just small clinical study using laser ablation, but yeah, I'm not sure if they look at those properties of, yeah.
I think correct me if I'm wrong, Chris. I think laser ablation is not common. I guess they're being used in investigational setting in patients. I don't think it's standard of care, so which I could explain why people have not looked into its influence in migration. I think people have started looking with the common treatment like temozolomide and radiation, how that affect migration, and that's being looked at.
Yeah, it's becoming more mainstream. Well, I shouldn't say mainstream. It's becoming much more common than it was even 2-3 years ago for the laser ablation. So the laser ablation, for those of you guys don't know, it's basically just a catheter or a probe with just a couple laser elements on a tip, basically just as thermal therapy. It's done under MR guidance, so you can actually see the sort of the map of the tissue that you're ablating in real time. So we actually do more ablation than most people around, because Clark Chen, who's the chair of neurosurgery here, is one of the early adapters of the therapy. It's previously used predominantly with brain metastases, but now they're starting to use it a lot more for unresectable glioblastoma in the upfront setting. And then we do adjuvant chemotherapy, radiation therapy with concurrent Temodar. But to get back to your question, I don't think anyone's really looked at actual cellular responses to there, because I think the thinking is, well, it just causes a bunch of necrosis. And then how does it actually change in the cellular mechanisms? So I've got a real quick question, Pekka. So for example, like when we do, when my IR colleagues will like thermal or cryoablate liver metastases, one of the biggest challenge is the location of the tumor next to vasculature, because then your vasculature can act like basically a heat sink or cold sink, depending upon if you're doing thermal or cryoablation. So when you're miniaturizing your probes, then how do you account for sort of the microvascular effects then too?
Yeah, correct. So for my current modeling, I'm just using PNS bioheat transfer. So that is assuming like no big vasculature around my tumor that is acting like as a tumor heat sink. But in the future, in my future modeling, I can use. There are some predictive modeling that you can just give the size of the vasculature and the proximity to the tumor to it, and it can just give you the effect that it might have on the process of the ablation. But we can also, if it's a big heat sink, we can also take that into account in my modeling as a like I can just take the convection of the blood into the account in my modeling to just take it into account. Yeah. Does that answer your question?
Yeah, it might be I guess it's tricky if the vasculature is like, yeah, I think maybe one thing about Chris's question is the vasculature in principle, I think volumetrically, the blood might be much bigger than the probe, certainly the blood flow. So it might be easier to account for via some sort of boundary condition or something, but I don't, you would have to kind of have a sense for what that is, which kind of gets to Chris's question, I think. How you would build that boundary condition, or model it or whatever. I don't know. Anyway.
Yeah, I don't have an answer to that right now, but yeah, I think there are predictive models that we can take the blood perfusion into account.
So is anyone planning to do the competition?
I'm probably not gonna do it just because of timeline, just because I'm trying to leave at the end of the year. So, but that definitely could be an opportunity. I think with Minhan and Baga, with collaboration with Dave's lab, there are a few students who might be interested in such work. And I can connect you if you're actually interested. I can connect you to people who does the Brownian dynamic modeling, and Minhan, I can connect you with people who does the imaging of tumors. I mean, Kellen, you already know the entire lab, and so you're really collaborating with us. So it might be worth writing something since you're already doing it anyway. Just kind of move it toward, immune stuff as well. I think the immune stuff will be really interesting. Glioblastoma will be worth exploring, and just if there is databases out there, you might be able to use such databases.
Right. Yeah. If the databases exist, it could be a good way to bolster the data that we are able to acquire as one singular institution. But.
Sure.
Okay. Well, good session, guys. Good conversation. I enjoyed the talks. So thanks for doing it.
Yeah. Thanks, everyone, as well.
Yeah, thank you.
All right. Have a good Tuesday.
Bye.